I'm a PhD student in the Health Sciences and Technology program at Harvard MIT. I'm a part of Chris Chen's research group at Boston University and the Wyss Institute at Harvard. It's my pleasure to introduce our keynote speaker for this year's Materials Day, Dr. Jennifer Lewis. Dr. Lewis is the Zhi-Ming Yu Professor of Arts and Sciences at Harvard School of Engineering and Applied Sciences and the Hans-Georg Wyss Professor of Biologically Inspired Engineering at Harvard. She has been a pioneer in the fields of additive manufacturing and assembly of soft, functional, structural, and biological materials. Prior to joining Harvard, Dr. Lewis was a faculty member in the Materials Science and Engineering Department at the University of Illinois at Urbana-Champaign. She earned her Doctorate in Science from MIT in Ceramics Science. Dr. Lewis has received numerous awards, including the National Science Foundation Presidential Faculty Fellow Award, the American Chemical Society Langmuir Lecture Award, and the Materials Research Society Medal Award, just to name a few. She is also a member of the National Academy of Engineering, the National Academy of Inventors, and the American Academy of Arts and Sciences, as well as a fellow of the Material Research Society, American Chemical Society, and the American Physical Society. She sits on the editorial board of Advanced Functional Materials and Soft Matter. She's also co-founded 2 companies that she's from her lab. Please join me in welcoming Dr. Lewis as she presents her work on in vitro vascularization of organoid building blocks in 3D tissues.
Thank you so much, Shoshana. Okay. Well, let me begin by thanking Alice White and Chris Chen for all the hard work that they've done to put together today's symposium. We heard already some amazing talks, and I hope to be able to contribute as well. So today I'm going to focus on, as Shoshana said, vascularizing organ building blocks in functional human tissues via biomanufacturing. But I think as Jason's talk really highlighted this morning, it's not just top-down fabrication that we need to leverage to do this, to achieve our goals. We really want to harness both biology and biomanufacturing. So Ken just did a great job of introducing all the complexity that tissue architectures require if we want to create and fabricate, design these. Shulamit's talk, of course, really highlighted the need for pervasive vasculature and ways and strategies in which to incorporate that both in vitro and in vivo. Trina talked about extracellular matrices, as well as did Jason Burdick in the first part of his talk. Of course, cells are a key ingredient in being able to replicate the complex architecture. And I think at the end of Jason's talk, he really pointed to the new direction and emerging direction in the field, which is to try to use multicellular aggregates as organ building blocks. And these could be in the form of cell spheroids, they could be in the form of organoids. And one of the things I want to pick up on from that is how do we design better organ building blocks, and how do we reliably fabricate them at scale to address some of the pressing challenges in tissue and organ engineering. But you know, that's just one part. I mean, what the tissue architecture requires, but also in order to do any kind of work in this field and do it well, I think there are also some other key ingredients. One has to have an innovative and talented research team, and I'll talk about that in just a moment. As I said, I think the platform needs to be able to harness both biofabrication and biology in an integrated way. And I want to also really point out a driving goal is to have tissues that both have form and function. It's not enough to just be able to print scaffolding in the shape of an organ. We need to be able to have functional tissue response. So let me start with the team. Usually this slide comes at the end, but as Shoshana mentioned, I am a material scientist by training. I came to Harvard about 7 years ago. And, you know, I had never done anything with cells in our lab. So needed to have a group of people to rally around the vision and really carry out the work. And the founding team shown here has done a tremendous amount of work over the past 7 years. And not only have they been just, you know, killing it in the lab, I want to say that they've contributed in a very substantive way to the ideas that I'll present today. So they've been really creative and innovative, not only hardworking. And as the time has gone on, we've added more people to the team that are also doing really exciting work. But today I'm really going to highlight the work coming out of this foundational team. So I think we all appreciate that there's an urgent need for renewable sources of human tissue, both for use in microphysiological systems, the so-called organ-on-chip models that can be used for personalized medicine, for fail-fast phase 0 trials for drug screening, and even for disease modeling. But equally important, I think, and yet a longer-term goal is to how do we can we make these in terms of autologous formats to alleviate the ever-widening donor supply and demand gap. And if we're using cells from you, made by you, and fabricating tissues for you, hopefully the idea is that we can reduce rejection after transplantation and alleviate the need for people to be on long-term immunosuppressants for the rest of their lives. So our platform, as I said, really we wanted to integrate both top-down fabrication, biofabrication, with bottom-up stem cell biology and harness what biological cells and assemblies of cells can do on their own. So we originally started with a process that we call multi-material 3D bioprinting, where we were printing cells embedded in hydrogel and co-printing alongside of that sacrificial inks that could be removed to create endothelial channels. And while we can create some very nice multicellular tissues that do in fact contain vasculature, we quickly realized that if we want to go after the kinds of organs that are in dire need, like the kidney, the liver, the heart, it's going to be important to be able to have architecture that spans many length scales, and I'll describe that in a moment. And in order to do that, and to do that in a scalable way, we also need to embed vasculature so that we can perfuse these tissues and sustain them in vitro before they're implanted. Now, the beauty of a lot of the emerging work in stem cell biology is that biology will kind of make these little mini organs or organoids on their own. As long as you provide controlled differentiation cues, they will self-organize into these multicellular assemblies with quite complex architectures. And I'll describe that in the first part of my talk. So in addition to harnessing biology, though, we do need to have some top-down control. So one of the key components of our tissues are extracellular matrix. And we've been working with a dual enzymatically cross-linked hydrogel that's composed of gelatin and fibrinogen, or fibrin, after it's been chemically, enzymatically cross-linked with thrombin. And we call this, for short, Gelbrin. So you might hear me use that term throughout the talk. We've also developed multiple sacrificial inks, but this was sort of our first initial foray into that. And this is just a triblock copolymer that has a novel response such that at room temperature, it's a viscoelastic gel and printable in a tubular kind of architecture. But when you cool it down, essentially the hydrophobic moieties become solubilized at low temperatures, leading to a fluid formation. And this fluid can be essentially washed away from the channels, leaving behind open conduits. Those conduits, of course, could be lined with endothelial cells or epithelial cells. And I won't go into this detail too much, but all of the inks that we want to print with, we tailor in a controlled viscoelastic response and a shear thinning response. So this allows them to flow very easily through the nozzles, much like Jason Burdick showed in his talk. They have this shear thinning response. The higher you essentially apply a pressure, the more easily the material flows. But when they exit the nozzle, in order to maintain this filamentary shape and prevent wetting and spreading, they rapidly re-solidify and have an elastic-like response. So this is why we use this term viscoelastic. So if we look at some of the work that we've done over the past several years on developing multi-material bioprinting methods, the first work that I want to show here was an example of thick tissue printing. So this was our first foray into printing architectures that were a centimeter thick, formed of stem cells. And what you can see being printed here is this fugitive ink in pink coming out of plane, but it's also integrated in 3 dimensions throughout the structure. And then the clear ink is a Gelbrin that contains stem cells. And we can then perfuse this entire tissue structure and sustain it for several weeks at a time. A second process that we developed was called embedded 3D printing. And this is some very early work from 2011 before we were even working with cells in our lab, we had a hydrogel matrix, and we were printing that same sacrificial ink inside this matrix. And we realized very quickly that if we could add cells, we'd have, a very nice vascularized tissue. And just to show you this idea of how this fugitive ink can be removed, this is really the seminal work that started off our multi-material bioprinting. You can see this sort of zigzag pattern that I showed earlier that we line with endothelial cells. This was originally printed with the fugitive ink, and then once we cool the chip down, we liquefy that and we can open up all of the channels. And this allows us to perfuse any of the tissue constructs that you see here, whether it be by direct biomaterial printing or by embedded bioprinting. So what I'd like to talk about then are two key advances from the lab. One is focused on kidney organoids. Primarily to date, we've been focusing on using these for drug testing and disease modeling, but we have obviously long-term goals around kidney engineering. And the second one is to focus on cardiac tissue engineering, and this is actually work supported in part by CellMet. I'm very fortunate to be a part of the NSF ERC at Boston University. And this builds on this technique that Mark and Seb coined as SWIFT, which is sacrificial writing in functional tissue. And this is basically taking embryoid bodies, assembling a tissue, and being able to directly write vasculature within them. So if we look at kidneys, they're very complex. We each have two of them. They serve very important functions. They filter our blood. And in the proximal tubule section, they also do a resorption process, which basically helps bring back some of the nutrients that have been filtered out back into our bloodstream. If we look at kidneys, they have a tremendous length scale and complexity. So they're starting from the filtration units here, these podocytes that form slit diaphragms wrapping the capillaries that are really responsible for filtration, to the capillaries that wrap around the tubules that the nutrients are reabsorbed back into, to the glomeruli that essentially have this kind of component wrapped in a Bowman's capsule, to, of course, a larger scale vascularized moiety. Kidneys have over 20 different cell types, and in our bodies we have 1 million nephrons per kidney. And these nephrons are the functional unit that does the filtration and reabsorption. Now, some of our early work had focused on just using bioprinting to create one segment of the nephron, which is the proximal tubule. And this is the segment that's very important for reabsorption. But as we did this work and we made the models more complex by introducing a vasculature by co-printing tubular architecture side by side, we realized that flow and 3D and microenvironments were really important. And that led us to think about, could we enhance the organ building blocks under flow to create building blocks that are not only vascularized at the micro scale, but also more mature, more functional? So I don't want to belabor this too much, but it really was the starting point for the work that we did in the kidney. And you can see that we're just basically here printing our fugitive sacrificial ink, if you will, on an ECM, which is then encapsulated by a top layer of ECM. Each of these individual printed features can be individually addressed, so we can then co-print alongside one another two open channels that are in a convoluted form, which is essentially mimicking the convoluted form of the proximal tubule. And we can line one of those with endothelial cells and the other channel with epithelial cells, proximal tubule epithelial cells, and we can really study the vectorial transport between these two channels, and we can study things like reabsorption. And so this is a set of images that show these two lumens side by side, co-located. We form both a confluent epithelium and endothelium, and as I'll show in the next slide, not only do we have the markers for functional response, like sodium, potassium ion transport, sodium glucose transport. These cells lay down their own basement membrane, but by putting them into this tubular architecture, into a 3D state and having flow, we see that these proximal tubule epithelial cells exhibit much better cell morphology and brush border, which is really where a lot of the reabsorption takes place. The cell border here at the interface, akin to almost what you would find in an adult human kidney. The cell height is about 75% of the cell height that you would find in a proximal tubule in a native environment. Microvilli length and microvilli density are also much higher in the 3D perfused on-chip case versus 2D same cells on our ECM, our Gelbrin, or 2D static on ECM, and of course, the worst case is just on a dish, on a plastic dish. So this gave us, quite a bit of insight into the role of flow on cell morphology in 3D microenvironments, but it turns out that that also played a very important role on their function. So these proximal tubule cells are responsible for reabsorbing glucose and delivering that back into the bloodstream. And we can see that again, that we see a pronounced enhancement in the function of these cells via the rate of glucose reabsorption in this 3D perfuse case, rather than 2D on a gel or 2D just on a transwell membrane. And this is roughly about 20% of the glucose uptake that you would expect for human kidneys. So if we come back to this picture, we were able to print, tubular architectures and vascular architectures, but being able to replicate the more complex filtration apparatus, the glomeruli and the podocytes that basically form foot processes and wrap around the capillaries, that's not something one can bioprint, in my opinion. And so that led us to think about how can we harness stem cell biology to recapitulate in these multicellular sort of mini organs, if you will, that mimic development. How can we create kidney organoids, and if we can make them, if we subject them to flow, does that enhance their function and vascularization? And so we were very fortunate to work with Ryuji Morizani and Naveen Gupta, who were at the time scientists in Joe Bonbancher's lab at Brigham and Women's, and of course Joe was a central part of this work too. And then two, I'll call them rock stars from my lab, Kim Homan and a graduate student, current graduate student, Kati Kroll, who really joined forces with Ryuji and Naveen to make this work happen. And Ryuji had published a beautiful protocol that's widely used to differentiate kidney organoids, starting with human pluripotent stem cells. And you can see that these organoids have many of the compartments that one would expect. They have glomeruli compartments here, which is shown by a podocyte staining the podocytes. They have proximal tubule segments. They have distal tubule segments. They even have endothelial progenitor cells. But what they lack is vascular network formation and maturity. They have the endothelial progenitor cells, but in the absence of flow, when they're cultured under static conditions, this vasculature just simply retracts. It recedes, it doesn't persist. And so we thought to ourselves, based on our very simple proximal tubule on a chip data, hey, why don't we subject these organoids to flow, and basically, would that help sustain vascular network formation? And so I guess the punchline is yes, it does. And we follow the Morizani protocol up to day 11 till we get to a pre-tubular aggregate state. Those organoids are in static culture up until that point, and then we put them down onto our ECM, onto our chip, and we can flow superfuse fluid media over those and continue their differentiation, but now under varying flow. And what you can see, first of all, here, when they're under static conditions, just being on the ECM that's adherent, this Gelbrin ECM, helps upregulate the vascularization. So we have staining here for MCAM and PCAM1, CD31 and CD146, and this is indicative of maturing microvessels within this individual organoid. Whereas if this same kidney organoid is seated on a non-adherent ECM, such as fibrin with or without collagen 1, again, there's some evidence initially of vessel formation, but as I said, over time that retracts and it's much lower in concentration. But if we add flow, amazing things happen. If we look at the upregulation of these FLIC1 positive, CD146 positive, and CD31 positive, which is basically a progression of immature to mature vascular cells. I mean, we see that under flow, it's a tremendous upregulation of these markers. And moreover, just visually, one can see the pervasive microvascular formation that occurs within these organoids compared to either their static counterparts on an adherent ECM or in a UL with no ECM. And it turns out that these vessels are partially perfusable. So if we look at this image, we're perfusing these fluorescent beads. Pretty soon this one you'll see will go out. We're only looking at now the channel, the fluorescent channel. And you can see that one of these vessels that is going into the organoid is actually able to have bead flow. And you can see also from this image that we do have some lumen formation within this microvascular network. So these are partly perfusable. And importantly, and I won't belabor this in too much detail, but these vessels do invade into the podocyte-rich regions, into these glomerular-like structures, and we see an upregulation of VEGF, which is a chemoattractant for this vascular invasion, and we see upregulation of nephrin and synaptopodin, which are functional markers of the podocytes within these features. So flow is not only helping to vascularize these individual compartments, but it's really helping to drive maturation and function of these features. And importantly, we see emergence of foot processes. So we see these podocytes starting to wrap around the capillaries that have migrated into and invaded into the podocyte-rich regions. And not only do we see that in the filtration unit in the glomeruli, but we also see enhanced function under flow for the proximal tubular components. So if we look at the tubular components, we can identify these cells by LTL marker, and we can see that this is located more pronounced in this apical region in the center part of this tubule rather than diffusely scattered under static flow. So this is basically what you would want to see in a more mature tubule. And again, we also see upregulation of many of the key markers and functional markers like aquaporin-1 for water transport, sodium potassium ATPase for ion transport, upregulation of the glucose transporter, and even sodium phosphate transport under flow conditions. So, So with that, we are certainly very interested in these vascularized kidney organoids as microphysiological models. There's a number of experiments that are going on in the lab right now in collaboration with others, folks at Roche, Uji's lab, to look at on and off target effects of different therapies, as well as disease modeling, for example, polycystic kidney disease. And we're finding that these vascularized organoids are much more able to recapitulate the kidney physiology compared to, say, static organoids. So we're making better building blocks. And I hope I've convinced you, at least from the images that I've shown so far and the comparison up here, that we're also able to recapitulate many of the sort of micro tissue architectures that one has that delivers function in the kidney. An overarching goal, of course, is to go beyond microphysiological systems and to create 3D kidney tissues for repair and replacement. And that's ongoing work. It's really in the infancy. But it's going to take advantage of some of the tools that I'm going to describe next, which is focus more on cardiac engineering. And so the next part of my talk, I'm going to highlight some work that we're doing based on Swift. And the idea here is, how do we take assemblies of these organ building blocks that have appropriate cell types and architectures and introduce vasculature now not at the micro scale, but at the macro scale. And as I said, this is done by using a variant, if you will, on 3D embedded printing rather than printing sacrificial inks in a hydrogel. We're printing them in a living matrix that are composed of these organ building blocks. They could be embryoid bodies, they could be pre-differentiated, they could be cell spheroids, much like we heard from Jason's beautiful talk. They could be differentiated organoids. So if we step back and we think about cardiac tissue engineering specifically, the goal of this is really to have patient-specific functional heart tissue, whether it be, smaller regions that can be used to heal a myocardial infarction, or, the ultimate big swing Westworld-like idea of having, a fully formed heart that can be used as a donor organ. I think that's the grand challenge, of course, and we can think about the steps to get there. First, of course, we need to have a digital model of this patient-specific organ, and those exist on NIH 3D print. They can be obtained for a given patient. There's been some beautiful work out of Adam Feinberg's lab. He calls this FRESH 2.0, and this was a paper that came out in Science, I believe in 2020, where he, showed that you can use embedded 3D printing, and this is more in a collagen kind of a matrix, to create a fully formed heart out of collagen scaffolding. He's also done some work to try to move down the field with respect to cardiac tissue using the same thing, but not at a full organ scale. And then Swift, I think, complements this work very nicely, because now we have cellular materials as the living matrix that can be vascularized and that can function. And ultimately, of course, the grand challenge meeting that goal is really going to require this integration of the capabilities of form with the capabilities of function. So if we think about all of the challenges in de novo engineering, we've got to have highly cellularly dense tissues. Most of these organs of interest have 100 billion cells at cell densities that are at least 100 million cells per milliliter. And as I showed with the kidney, many of the organs of interest have tens of different cell types. So right away we have a cell source, cell density challenge. We also have a complexity across length scales challenge. Much like we see for the kidney, we have the same kind of complexity in length scales in cardiac tissue. And then in order to do anything at scale, we need to have this embedded vasculature. And Shulamit made this point very nicely in her talk. It's the next hurdles in tissue engineering are both complexity across length scales and being able to create a tissue that's vascularized and can be sustained in vitro at scale prior to implantation. So if we think about how to approach that, fortunately, there's been a series of really great breakthroughs from others in the field, including the beautiful work for being able to harvest iPSCs, or harvest cells and transform them into induced pluripotent state, which can then be a renewable cell source that are patient-specific. If this patient has a disease, one can also use techniques like CRISPR to manipulate those cells and perhaps cut away, if you will, remove the disease of interest. We can then take advantage of all of the beautiful differentiation protocols, such as the one that Ryuji Morizani did for the kidney organoid building blocks, to develop these multicellular 3D tissues that have this tremendous diversity of cell types, as well as architectural features that really drive function. And then the idea is that with Swift, we can take these building blocks, create a big matrix of them, and write vasculature within them. So I've highlighted Mark and Seb already, but I want to give a shout out to both of them. Mark is now an assistant professor at Stanford and bioengineering, and he's off to an incredible start there. And Seb Uzel is on the job market looking for his faculty position, ready to start his lab. And so together they formed the Swift team, and they really kind of pioneered this whole idea of starting with these multicellular building blocks, creating a living matrix, and then printing a vasculature within them. And I'm just showing the end of one of the build sequences here, but you can see that we have the ability to perfuse these tissues, and I'll show that to you later. But the idea is that once we pattern the sacrificial ink within this living matrix, we can remove it to open lumens like I showed before. Then we can perfuse these channels to sustain this highly cellular dense tissue. The organ building blocks will fuse together, and again, Jason showed very nicely in his talk that this can happen even when they're not perfectly packed adjacent to one another. And then, of course, we need to characterize tissue function. So let me first show you this movie here, where what you're seeing are these OBBs, these organ building blocks, jammed into a matrix. And now I'm just translating a nozzle, an empty nozzle, nothing's being printed through, and I'm just showing you that these building blocks self-heal around behind the nozzle. And now I'm translating the same nozzle through, we're imaging the tissue from below, and here is the sacrificial ink being printed. Now you might ask yourself, if these building blocks are so mobile that they can move around and reform behind the nozzle, how do we actually remove the sacrificial ink without basically collapsing these organ building blocks around that printed lumen? And I should point out that this tissue is about 2.5 milliliters of these embryonic bodies, which means roughly that translates into 400,000 or OBBs, or embryonic bodies in this case, or a total of roughly 500 million cells. So this was a heavy lift just to even create the number of building blocks needed. So what's the secret sauce? How does this work? How are we able to pattern this ink seamlessly through the self-healing matrix, but then allow the lumens to be patent and perfusable after we've created the tissue? Well, this is where Seb and Mark really teamed together to come up with this idea of having an extracellular matrix that under the printing conditions, so here now we're printing at low temperatures and we're removing the ink at elevated temperatures. It's the exact opposite of the first part of the talk. But they developed this ink that's collagen-based that essentially doesn't contribute anything to the rheology of the living matrix during the sacrificial printing process. But after the tissue is heated up to 37 degrees C, that extracellular matrix solidifies, it undergoes a gelation process, and that holds all of the little building blocks together. And then we can remove the sacrificial ink at elevated temperatures because it is actually liquefying at 37 degrees C. So that's kind of the secret sauce behind Swift. So I hope I don't have to convince you, but if you create such a cellularly dense tissue at sort of approaching centimeter scale, without vessels in it, the cells are going to die. And so this is an example of a 4 by 4 by 4 millimeter tissue construct, again, with cell densities of 200 million cells per milliliter. And in the core, it's completely necrotic. All of these cells have died. The tissue thickness viability region is less than a millimeter thick here. If we then introduce that vasculature within the tissue matrix, and these are slices taken through a Swift printed embedded vascular system, again, same high cell density, we can get essentially near full viability of cells across that tissue architecture because of the ability to perfuse through the embedded channels. And by just varying the printing pressure through an individual nozzle, we can vary the lumen size on the fly during printing. So we can create these branching architectures that have different diameter channels as we're printing by just simply changing the pressure. Now, Jason pointed out this idea of pick and place. So he was using a print head essentially to pick and place organoids one by one. Much like you pick and place surface mount components on a circuit board, on your printed circuit boards, if you opened up your computer. That was a very elegant technique, but one can also do Swift printing of organoids in organoids. So this is an example of two different types of organoids, and Seb carried out this work where he was printing fluorescently labeled organoids, in this case embryonic bodies, pre-differentiated inside a living matrix of non-fluorescently labeled embryonic bodies. So you can imagine starting to think about answering some of the questions that Jason is doing so elegantly, like if we were able to create these sort of scarred, if you will, fibroblast-rich organoids and print them into a three-dimensional construct, we could really create a myocardial infarction that's reminiscent of what you would experience during a heart attack in a bulk three-dimensional tissue. So it's very exciting to think about co-printing different types of organ building blocks alongside of one another, along with vasculature. And again, these are millimeter volumes of tissue. So the beautiful thing about Swift is it works with any organ building block. Essentially, we can have embryonic bodies that are pre-differentiated, as I've said. A lot of the initial work we did was with EBs. We can have different types of organoids, even cerebral organoids, which is one of the areas of interest for Mark Schuyler Scott. And we can make these cardiac spheroids. So these are multicellular aggregates akin to what Jason was describing. We follow a protocol from Sean Polasik's group at Wisconsin to make these at very large quantities. As I said, we're talking about milliliters to a few milliliters of tissue. We follow this protocol to create iPSC-derived cardiomyocytes, and we can combine those, at least initially in this work, with fibroblast cells. You can see that the cell density is quite high. It's exceeding 200 million cells per milliliter, and the total cardiomyocyte concentration is on the order of 200 million cells per milliliter. And these individual building blocks beat. They are not beating synchronously, but they are asynchronously beating. And this is an image taken 9 days after the post-differentiation process. So this is essentially our living matrix. These organ building blocks are what constitutes the Swift living matrix. Once we've assembled that living matrix, as I've shown before, we can print vasculature within it. And this is just one branching architecture. And after removing the fugitive ink, you can see this dye being flowed through the channels. So we have the ability to introduce vascular features. We can then look at the matrix on day one, as you know, after we've introduced the Swift channels. You can see that these building blocks have not yet fused, and the tissue is not beating synchronously. Each one of these is more or less acting still like an individual organ building block. But by day 10, the tissue is fused and it's beating more synchronously, and I'll show you a calcium imaging to confirm that. And we see the evidence of the sarcomeric machinery that you would expect within the cardiac tissue. But I want to say that if we make these building blocks even more cellularly complex, we can enhance the contractility quite significantly. So in the last few slides, I want to show first this calcium image mapping taken first at day 1, where we have asynchronous beating. And this was from regions 1 and 2 that were sort of the opposite sides of this tissue construct. By day 7, you see that both sides are beating synchronously. So we do get tissue fusion and we do get synchronous beating. And then we can paste these tissues as shown here. We can use these for cardioeffective drug response. And you can see that in the presence of isoproterenol that we get an enhancement in the beating frequency. And with peptinol, we actually suppress that. And that's what you would expect for these cardio-based drugs. But lastly, I guess with the time remaining, I just want to highlight work that is not published that basically enhances the complexity of these cell spheroids, these cardiac spheroids. And this is work that Seb Uzel is leading, along with a collaboration with Chris Chen's lab. And the idea here is, instead of just having iPS-derived cardiomyocytes and fibroblast cells, what happens if we introduce HUVECs? And I will show you that the tissue contractility significantly enhances. So this is day 8 after differentiation, where now the cardiac OBBs have fused into a beautiful synchronously beating tissue. And if we look at this tissue, we can see the presence of lectin-labeled endothelial cells. So in fact, we do have, much like I showed with the kidney organoids, we now have the ability to incorporate microvascular features into these building blocks, and we see a direct impact on their function, on tissue function. And working with Greco Song, who just fairly recently defended his PhD within Chris Chen's group. We've been very fortunate to work with them to take some of our Swift-based cardiac tissue and start to begin to look at animal studies. This was implanted in the fat pad of a nude mouse, and you can see day 14 post-implantation with two different types of extracellular matrix that we use in the Swift printing process. The original matrix, collagen 1 rich, and a new matrix that we've been working on, a new ECM for Swift that's fibrin rich. And you can see that there's much better vascularization with this second generation Swift ECM. And if we introduce a dextran, a TRITSI labeled dextran, we can see that there is integration of the Swift tissue with the surrounding host vasculature is integrating into the tissue. And so this is very early work, but we're super excited to see where this goes. So back to sort of the ingredients that we need. The last thing is if we want to try to, take Swift and do something with patient CT data, just to convince you that that can be done. This is a tissue wedge taken from 3D NIH patient data. And this is a wedge of tissue that is filled with these cardiac OBBs where we're introducing the printed vasculature. And this is what that sample looks like. So that's kind of the roadmap of where we're going. I'll end by, again, thanking you all for your attention, and the organizers, Alice and Chris, for the kind invitation. Our kidney work was supported largely by NIDDK and NCATS, as well as Roche. The work on the cardiac Swift was generously supported by a DoD Vannevar Bush Fellowship, which is like blue sky money and really we needed a lot of blue sky money for that work. And also, as I said, we're very fortunate to be a part of the VU ERC, the NSF ERC on CellMet. And I've highlighted numerous times, I hope, the incredibly valuable contributions of collaborators and members of the team. And I just want to say we are looking for postdocs, so if this sounds exciting to you, please reach out. So with that, I think I'll stop sharing and come back and take any questions that you might have. Hopefully I haven't lost you guys. Okay. I see.
Yeah, you're all set.
Okay, great. It's always, when you're talking to a screen, you never know.
Buddy.
Yes, exactly.
All right, we'll go ahead and take any questions for Professor Lewis. Okay, first question is, how big are the cardiac organoids, and is there a minimum
that's a
size?
great question. In this first work, they were 400 microns in diameter. We've worked with organoids as small as 200 microns. The minimum size is going to be set by what is the architectural features that you need to replicate in the building block itself. And so, for example, in the kidney organoid work, once you go below a certain size, like say 100 microns, you may not have very many nephron units. So there is a reasonable minimum size. At the same time, you might imagine that the smaller the building blocks, the smaller the vascular features that one can print in. So there is some motivation to optimize that size to recapitulate function, but also be able to have multiscale vasculature. So with these organ building blocks, with the Swift building blocks, we were printing channels anywhere from like 400 microns in diameter, so akin to the building block size itself, up to 1 millimeter. And of course, you can go actually higher than that. So great question. Thank you.
Okay, the next question is, I'm curious whether you envision more classes of materials that need to be developed push the limits of fugitive ink usage or other limitations that need addressing?
So I think if I heard you right, the question is more materials development. Is that right?
Yeah, whether you envision of materials that need to be developed to push the limits.
Right. Okay. Thank you. Well, we've actually shown 2 fugitive inks in this talk. So the pluronic-based one for the direct printing and a gelatin-based ink for the swift printing. As for the ECM, I think we're obviously still refining the extracellular matrix for swift, and we've seen significant effect of the ECM. So by no means is that a done deal, completely fully optimized. So there's more work to be done there. The Gelbrin material that I described earlier is actually quite well suited for the kidney engineering work that we're doing. So perhaps there we're very happy with the ECM. But obviously there's a very large body of literature on tailoring extracellular matrices that I'm sure we could benefit from harnessing more of those advances. So, yeah.
Okay, the next question is, is there any in vivo work going on trying to utilize the vascular nephron organoids that you got under perfusion?
Yes, so we are not yet starting in vivo there, but it is on the horizon. So thank you for that question. We have not yet started that work. Others have, Melissa Little, and there are others in the field that have implanted non-vascularized organoids, so organoids under static culture, and they've showed that they do integrate with the host, and much like I showed with the cardiac samples with Chris Chen's lab, that native vasculature will come in and invade and integrate. But we haven't actually done the vascularized organoid in vivo work yet. It's a high priority.
All right, thank you. We have time for 1 or 2 more questions if anyone wants to submit. Okay, next question. Would the vasculature get squished/deformed when tissue constructs grow and swell along with time?
That's a great question. So yeah, I would say from the perspective of what we've seen so far, these tissues, as they mature and fuse, of course there is some contraction. So you can imagine that at least initially there's some reduction in the vascular channel diameter just because the tissue is fusing. We haven't, done the kinds of implantation models for long term to see if there's growth and maturation that way. So I would say that's an open question. I don't know the answer yet.
Okay, the next question is, given that cardiomyocytes are highly aligned in the cardiac muscle, can SWIFT be used print aligned structures?
Yeah, I'll just say stay tuned on that. We are working in that direction.
Okay, next question is, how do you create flow on vascularization of organoids?
That's another ongoing aspect of the work, and Katharina Kroll is leading that effort, but we are basically using some of our organ on chip technology to promote the hookup between the embedded microvasculature within these organoids and printed vasculature. And that work is looking very promising, I must say, but we have not yet published that. But the idea there is ultimately to be able to perfuse through the printed vasculature and then go through the microvasculature and the organoids and essentially out to another printed channel. As you know, the beautiful work of Chris Chen, where he's doing angiogenesis between 2 sort of pattern channels, has shown very nicely that one can do that through capillaries that grow and form between 2 co-located channels. The challenge is, of course, to do that now in a 3D multicellular tissue like these organoids, and that's something that we're working really hard to do.
Okay, thank you so much, Professor Lewis. We'll move on to the next.
Okay, thank you.